Hi, my name is Gilda Bocco.
And I'm Berchick.
And we'd like to talk to you about our project, the analysis of unknown gene functions in the bacteriophage NormanBulbieJr. So first off, a little bit of background. So NormanBulbieJr was a cycloviridae phage that we actually were able to discover here at USciences in 2017. We classified it as a F1 phage, and it's known to be a tempered phage, which was proven via the integrase found on the genome, which is gene 48 here. And this is an EM picture of NormanBulbieJr for reference. And overall, this genome consists of 102 genes. Our primary focus of this project is actually to be able to get a better understanding of the genes with unknown functions within this genome, and then be able to use that information to help other genomes within its own database to be able to help other scientists use this information. And this can overall help for future applications such as antibiotic resistance and being able to engineer a phage with the right genes to be able to treat those infections and those strains. So we are currently studying 25 different genes of interest within the genome, but we're going to talk about two when we're talking about results. So the first one is NormanBulbieJr gene 25. I chose this gene because it currently had no known function, and it is the only reverse gene in a sea of forward genes, which I find to be very interesting. And its overall length is around 190 base pairs.
And I chose gene 18. It is a minor tail protein, and I wanted to see if it actually was a minor tail protein. So hopefully doing more research into this gene will help me to prove its true function, and its overall length is about 2,500 base pairs. So these are the materials and methods we've been using over the course of this semester. We are currently at stage 2, which is investigating gene-dependent phenotypes in the host. So we've been doing a lot of molecular cloning. We successfully cloned and confirmed our gene of interest into P.extra, and we're currently working on transforming this gene of interest into the host organism M. smegmatis. From there, we can perform phenotypic assays like the cytotoxicity assays and defense assays.
So here are results that we were talking about a little bit earlier, and these are going to be the two genes that we're focusing on. So the first one is gene 25, and as you can see here, the first one is PCR amplification. That's where we were able to correctly identify if we're amplifying the right gene. And the amplicon size that we were looking for, and the one that was produced here in this figure, is around 250. We were then able to insert that gene of interest into the plasmid P.extra, which we can confirm via the presence of these transformants. And then we were able to successfully get that plasmid into the E. coli, but then we need to make sure it contained that gene of interest. So we were able to do clone verification, and that's what you see here in the last picture via PCR, and the amplicon size that we were looking for was around 300 base pairs, and that's what we saw produced here.
So similarly, for what Gilda was talking about, for gene 18, we did PCR transformation and clone verification. For PCR, with the gene-specific primers, we generated a band around 2,600 base pairs, which is what you see on the left. And then we transformed that gene of interest into E. coli and plated it. So the presence of colonies means that transformation was successful, but to test whether these colonies contained the plasmid with the gene of interest in it, we once again performed PCR, and it was producing colonies, or producing bands rather, around 2660 base pairs, which is what you see generated on the right.
So overall, this is all the genes. There are two different slides here. This is all the genes that we were currently working on within the class entirely. And as you can see here, we're at different steps for each of them. Only one of them right now is currently working on a cytotoxicity assay, so we're going to be able to get results for that soon. Many of us are transforming those genes into smegmatis currently. But we've got a lot of success amplifying and being able to get everything going so far. Here's the second slide, second half of the genes that we're working on. And then
so for future directions, we'll be transforming this plasmid containing our gene of interest from E. coli to M. smegmatis. And this will verify the presence of the gene of interest in the plasmid in the new bacterial host. And then we'll be conducting phenotypic assays like cytotoxicity assays and defense assays to determine the gene's possible function.
Here are references, and these are a few acknowledgements. Thank you to all the people listed on here, because without them, this class would not be possible.
Or as much fun.
Yeah, so. Exactly.
Thank you for watching.
Thank you for watching.